Welcome to Neutralizing COVID, the monoclonal antibody story, the podcast where we unpack the science, strategy, and real world impact of monoclonal antibodies in the fight against COVID 19. Thanks for tuning back in. I'm Dr. Shikha Agrawal, and in this episode, we'll spotlight a standout monoclonal antibody from the very early pandemic days. Join me for the antibody blueprint Sotrovimab stand against early COVID variants. I'm thrilled to have Dr. Aiswarya Chaudhuri, a nanomedicine expert, back with us for another deep dive today. Welcome to the show. It's time to peel back the layers of this fascinating topic.
Always a pleasure to join you. Thanks for inviting me back, Dr. Shikha.
So, to start, could you give our listeners a quick refresher on what monoclonal antibodies are and how they help us fight viral infections like COVID 19?
Certainly. Monoclonal antibodies are laboratory made proteins that mimic the immune system's ability to fight off harmful pathogens like viruses. Think of them as highly specialized hunter molecules. When you get a viral infection, your body naturally produces a variety of antibodies to attack the virus. Now, monoclonal antibodies are essentially a purified, concentrated dose of one specific, highly effective antibody that has been selected for its potent virus neutralizing capacities. They can be designed to target a specific part of the virus, like the spike protein of the SARS-CoV-2, to prevent it from entering the human cells and replicating.
That's a great explanation, Dr. Aishwarya. Sotrovimab made headlines as a promising treatment early in the pandemic. What was the unique about its origin and development?
Sotrovimab's story was indeed quite interesting. It was developed by the GlaxoSmithKline and Reed Biotechnology. One of the most remarkable aspects of its origin is that the parent antibody called the S309 was actually isolated from a survivor of the 2003 SARS outbreak, that's the SARS-CoV-1. The idea was that an antibody effective against SARS-CoV-1 might also be effective against SARS-CoV-2 given the similarity between these coronaviruses. This hinted at the possibility that S309 and subsequently Sotrovimab targeted a part of the virus that doesn't change much across different but related coronaviruses, what we call a conserved region.
Targeting a conserved region sounds like a key strategy, especially with viruses that mutate frequently. How was Sotrovimab engineered to enhance its effectiveness and achieve broad neutralization against those early variants?
That's right. The fact that S309 antibody can neutralize both the SARS-CoV-1 and SARS-CoV-2 was a strong indicator that it bound to a highly conserved epitope or the target site on the spike protein. Now, this is crucial because the target site is conserved and the antibody is more likely to remain effective even as the virus evolves and new variants emerge. Now, specifically, Sotrovimab was engineered in a couple of significant ways. Firstly, it was designed to bind to those conserved regions of the spike protein distinct from the more rapidly mutating receptor binding domain that many of other antibodies target. This gave it an edge in maintaining activity against various early stress. And secondly, it underwent what's known as FC engineering. Now, a specific modification, often referred to as an LS mutation, was introduced into the FC region, the antibody's tail. Now, this modification helps us to extend the antibody's half-life in the body, meaning it stays active for longer, and it was also thought to potentially improve its distribution into lung tissue where the virus does lots of its damage.
So it's not just about finding the right antibody, but also fine tuning it for better performance. Could you elaborate on how Sotrovimab actually worked to neutralize the virus once administered to a patient?
Sotrovimab works through a couple of primary mechanisms, and the most direct is neutralization. By binding to that critical conserved part of the spike protein, it physically blocks the virus from latching onto and entering the human cells. Now, if the virus can't get into the cell, it can't replicate and spread effectively. Now, beyond direct neutralization, sotrovimab was also designed to engage other parts of the immune system. Its FC region can interact with the immune cells, leading to a processes like antibody-dependent cellular cytotoxicity and antibody-dependent cellular phagocytosis. In simple terms, it means it will flag the virus-infected cells for destruction by the body's own killer cells or mark virus particles for engulfment and elimination by the phagocytic cells. So it's a multi-pronged attack.
Such an important clarification. Now, speaking of effectiveness, how effective was Sotrovimab against those early variants of COVID-19 like Alpha, Beta, and Delta, which caused so much concern?
Clinical trial data, such as the COMET ICE study, was very encouraging for its use in high risk individuals with mild to moderate COVID 19. The results also showed a significant reduction in the risk of hospitalization or death, around 79% in that particular trial compared to the placebo when given earlier in the course of infection. This was during a period when the variants like alpha, beta, gamma, and later the delta were prevalent. Now, real-world evidence studies also supported its effectiveness in reducing severe outcomes during waves dominated by these early variants. Its ability to retain activity against these different strains was a key advantage at that time.
That sounds incredibly promising. However, as the pandemic progressed, new variants like Omicron emerged. What challenges do rapidly evolving viruses pose to monoclonal antibody therapies like Sotrovimab?
This is the central challenge in developing the antiviral therapies, particularly the antibody-based ones. Now, viruses, especially the RNA viruses like the SARS-CoV-2, mutate frequently. While Sotrovimab was designed to target a conserved region, no region is completely immune to mutation. Now, as the new variants like the Omicron emerged with numerous mutations in their spike proteins, the effectiveness of many monoclonal antibodies, including the Sotrovimab, began to wane against some of these newer forms. Now, for instance, data showed that Sotrovimab had reduced activity against certain Omicron sub variants like BA2. This led to revision in its emergency use authorization in some regions as those sub-variants became dominant. It underscores a critical point. Now, the fight against evolving viruses is a dynamic one. An antibody that is highly effective today might be less so tomorrow if the virus changes its lock, the binding site of the antibodies.
So the virus essentially learns to evade the therapy. You mentioned Sotrovimab targets a conserved region. Why is this so important in antibody design, and what does its story teach us for future pandemic preparedness?
A great question, and you're absolutely right, Dr. Shikha. Viruses, especially these RNA viruses like the SARS-CoV-2, mutate rapidly, and that constant evolution can render antibody therapies ineffective if those antibodies target parts of the virus that frequently change. That's where the concept of targeting a conserved region becomes crucial. A conserved region is a part of the virus's structure that remains relatively unchanged across different variants because it's essential for the survival of the functioning of the virus. Now, Sotrovimab was designed to bind to such a region of the spike protein, specifically a site that tends to stay stable even as the virus mutates elsewhere. Now, this strategy not only helps the Sotrovimab retain effectiveness against multiple early variants, but it also teaches us a broader lesson for the future pandemic preparedness. When designing antibody therapies aiming for the conserved, functionally important regions can offer a more durable defense. It's a way to future-proof therapies against viral evolution, which will inevitably happen.
Excellent point about conserved regions being the virus's Achilles heel. It really puts into perspective both the incredibly ingenuity behind the therapies like Sotrovimab and the relentless challenge posed by viral evolution. Before we wrap up, it's important for our listeners to remember that our discussion today is for informational purposes only and is not a substitute for professional medical advice. Always consult a healthcare provider for any health concerns or before making any treatment decisions. Dr. Aishwarya, thank you so much for sharing your expertise with us today.
Glad I could be a part of this, Dr. Shikha.
That's a wrap for this episode. To our listeners, we appreciate you spending part of your day with us in neutralizing COVID, the monoclonal antibody story. If you enjoyed this episode, please subscribe and spread the word, and we'll catch you in the next one.